Good morning. Thanks for the ASM and Jiren for the invitation. And it's a great honor to be here. Today I'm going to talk about a small story, unpublished story about Vibrio Cholerae and how Vibrio Cholerae interact with gut microbiome. Okay, so probably you heard a lot about gut microbiome, right? So in healthy people, like you and me, we have very complex microbiome. And those are a lot of different bacteria or other microbes. And they provide a lot of benefit. And they particularly for those can fend off invading pathogens. So colonization resistance is pretty well studied. They have different mechanisms, right? So this is not the topic of today. But during different disease, many diseases, the microbiome changed, become diversity reduced, and those, for example, in long COVID, diabetes, malnutrition, and so on, so many. And they become very simplified microbiome. But the question is, like chicken egg, right? Which one caused which? Right? So I'm going to use Vibrio Cholerae as a model organism to tell you a story about those dysbiosis can affect cholera outcome. Okay, so Vibrio Cholerae. Vibrio Cholerae is a gram-negative bacteria. It's a highly motile, has this single beautiful flagella, and motility is critical for Vibrio Cholerae pathogenesis, and also is critical for my story today. And Vibrio Cholerae is causative agent for the disease. Cholera is acute intestinal infection. The symptoms involve massive diarrhea and vomiting. Cholera is still endemic in many parts of developing countries, usually associated with poor water, sanitation, and poverty. For example, in Bangladesh, there's over 100,000 cases a year. And there's many Bangladesh people at high risk for cholera. Okay? Recent studies show that for those Bangladesh people, even they are healthy Bangladesh people, their microbiota is kind of disrupted. And they have low diversity and dominated by Streptococcus, Enterococcus, and E. coli. Okay? And for regular people like us, we're pretty resistant to cholera. And those complex microbiome can inhibit virulence gene expression, and they use mouse model. You can see the colonization actually got inhibited. But how dysbiotic community impact cholera is not really known. This is our we try to study this. We ask the question is, how about those dysbiotic community can affect Vibrio Cholerae colonization? We use a mouse model, and you can see that when we have those dysbiotic community, E. coli, Enterococcus, several strands of Streptococcus, and when you present of those DS community, Vibrio Cholerae colonization increased. Okay? And because you don't always want to use mouse model, right? So we use an in vitro model, a tissue culture model, and you can see that the Vibrio Cholerae can attach those tissue culture cells, epithelial cell, but in the presence of DS bacteria, this Vibrio Cholerae is labeled green GLP labeled. So when you have DS, the number of Vibrio Cholerae attached to the surface increase. More importantly, when we use the supernatant span medium, that you get rid of cell, only the supernatant also can induce Vibrio Cholerae attached to the epithelial cell surface. So this suggests those DS cells, bacteria can produce stuff that secrete out and in the cultural medium. Okay, so then we try to see how this affects Vibrio Cholerae, right? And we use this microfluidic device. You can see that Vibrio Cholerae go in this way, and this way go DS supernatant or medium control. Sorry, use touch screen, no? Where's the mouse? No, click that. Okay, great, thank you. So you can see that many bacteria, Vibrio Cholerae, go into that direction. We show it's actually not chemotax, it's indeed just motility, increased motility. You can see that when you have DS, without DS, Vibrio Cholerae alone, they're fine. They calmly swim around. But when you have DS supernatant, they suddenly get very excited. They're just running really fast. The average speed increased dramatically. Okay. So then next one, we will, of course, before we use the community. Now we just separate those to see which one. And we found it's not E. coli, it's not Enterococcus, but it's Streptococcus. Different strands of Streptococcus all can enhance Vibrio Cholerae colonization. And they also enhance Vibrio Cholerae motility, those highlighted one, enhance motility and adherence. But when we test other Streptococcus species, they also can enhance. All the one we tested, those green, labeled green, they can all increase Vibrio Cholerae motility and adherence, right? So we kind of universal. We tried to do biochemical study, and long story short, and we think it's some kind of peptide. It's a quorum-sensing peptide. It's in gram-positive bacteria. They use the quorum-sensing molecule is peptide. They went at high cell density. Those peptides can interact with certain response regulators and activate gene expression. But in Streptococcus, this is a pyogenes, and in many other Streptococcus, they have those peptides called short hydrophobic peptide, SHP, and they at high cell density, they can bind to the RGG transcriptional regulator and activate the transcription, different gene expression, majority biofilm formation, and other pathogenesis-related genes. Okay, so those peptides are mature, after process become mature peptide, it's only 8 amino acids. For example, pyogenes is DIILIVGG, right? So strep, infantarium, the one in the DS community, very similar GIIVGG, right? So we synthesize those peptides, those synthetic peptides, and all these peptides can induce Vibrio Cholerae hypermotility and can induce Vibrio Cholerae adherence to the epithelial cells, right? So all these peptides work. So it's peptide that did the trick. And furthermore, we make deletion of SHP in one of the DS strands, Streptococcus infantarium, and the delete of this SHP gene, and this strand no longer responds, no longer make Vibrio Cholerae become hypermotility, and then no longer induce Vibrio Cholerae attachment to the epithelial cells. And more importantly, it no longer enhance Vibrio Cholerae colonization of infant mouse. Okay, so we think those peptides lead to the promotion of Vibrio Cholerae motility, and then subsequently enhancing attachment and in vivo colonization. And we named SVIPs, right? Streptococcus virulence inducing peptides. And we want to find why, right? How it induced, not why, sorry, how. Then we test different strains, different bacterial strains. We found all these SVIP can induce motility of different Vibrio Cholerae, and other Vibrio species, and also Bacillus subtilis, but not E. coli, not Salmonella and Pseudomonas. So what's the difference between these two sets of bacteria? And it's actually the status different. And they all have all the similar flagella structure, but because in order for flagella to rotate, they need to use the generally iron motive force, and this status is this kind of ion channel, right? And but for multi-AB like E. coli, they use proton channel, proton force, and for PALM-AB in those Vibrio, they use sodium force. So they generate different sodium motive force and to rotate the flagellum. So it looks like it's probably affect sodium concentration, right? And we use the dye that can bind sodium, intracellular sodium, and then become fluorescent. Vibrio Cholerae labeled, M-cherry labeled Vibrio Cholerae, but when we add SVIP, those peptides become strong, fluorescent become much stronger, and they increase a lot. So it's indicating that the intracellular sodium concentration was increased when you have those peptides. Okay. And then this AlphaFold, right? AlphaFold can predict the interaction between the peptide and those PALM-AB. We found this is interact with C-terminal of PALM-A. And those PALM-A, like 6 subunits of PALM-A and 2 subunits of PALM-B, right? This is form this ion channel, sodium channel. Okay. So then we just delete, not delete, we make point mutation of this critical amino acid. That one is supposed to interact with sodium, sodium ion. And you can see that for Y type, if you add SVIP, the speed increased. But for this mutant, they still multi, but no longer responds to SVIP. Okay. Then final evidence is that we overexpress PALM-AB, Vibrio Cholerae PALM-AB in E. coli. Remember, E. coli is proton-driven force. They don't have sodium ion channel. And so when you have PALM-AB plus SVIP, this measure the fluorescent sodium concentration, intracellular sodium concentration, look like it greatly increased when you have PALM-AB and SVIP. This suggests that the SVIP directly interact with PALM-AB. Okay, so conclusion is that we have those streptococcus gram-positive bacteria. Gram-positive bacteria, quorum-sensing peptide in the dysbiotic community can drive cholera pathogenesis by interacting with flagella status and increasing sodium motive force. Okay, that's all done in vitro, in the lab, and in animal model. So what happened in human, right? So previously, people want to study how those pre-colonization, pre-infection colonization, basically microbiota, how those can affect Vibrio Cholerae infection and disease severity. But it is not that easy that you can just collect microbiome, right? You collect the fecal samples. Because when patients went to the hospital, they already have diarrhea. So all those microbiota are already wiped out. So Anna Wells, she's from the University of Washington. She did a very smart work that when you have a patient come in, and then they just go back to their house, look for people, the household contact, right? They collect samples, fecal samples from those people, and then monitor them to see who will get infected, who will get the disease. So many people actually is pretty resistant, uninfected, or for those infected, that you can detect Vibrio Cholerae, but then there's no diarrhea. And for those, or some called asymptomatic and less severe, right? For symptomatic, it is just regular diarrhea. Then they go back to the sequenced one. They can sequence a metagenomic sequence for those collected in day one, okay? So then you know the pre-infection microbiota. Right? That's pretty smart, I think. Then we approach them to ask them to reanalyze their data. They have published data 3 years ago. And they specifically look for streptococcus. Okay, we see that in asymptomatic, the relative abundance of streptococcus in symptomatic patient, those pre-infection microbiota is increased. Okay, so it's not after it's virtually all wiped out. So pre-infection microbiota, the relative abundance increase in symptomatic one. And then look specifically for the RGG SHP binding sequence, and we also see the increase, relative abundance increase in symptomatic. So this is really important. Here, when we look for different species, those when they use orange colors, those have significance. They all mostly increase, okay? So this really suggests that if you have some streptococcus, you are more prone to get cholera. Okay? So this is a working model. We have this DS community, and they produce short hydrophobic peptide, SVIP, and they do their own business, right? They use quorum sensing to regulate their own gene, probably promote them to make better colonization and so on. But when Vibrio Cholerae come in, Vibrio Cholerae can sense the SVIP and come fast mode, reach to the intestinal surface faster. So when I make this model, my daughter, she's a college student, thought, oh, such boring model. Okay? Then she quickly spent like 1 hour and made this drawing. She thought this streptococcus can make a flying magic carpet for Vibrio Cholerae. She even decorate those peptide sequence there and make Vibrio Cholerae much stronger. So that's why the title comes from. Okay, but actually it's not just Vibrio Cholerae. We think that we did use a community isolated from Bangladesh, and in fact, germ-free mice together with either wild-type streptococcus or SHP streptococcus. And for 3 different communities, the outcome is completely different. And but between wild-type and SHP are different. Many of those species that's not even multi. So this indicates that probably peptides can control more than its own quorum sensing, and also more than just motility of Vibrio, probably can control many other things. That's a way for us to study further. Okay, so my lab is in beautiful Philadelphia, and the work majorly done by talented postdoc Polly Poo, and with help with many other people in the lab. The collaborators here, and funding. Thank you so much.
Any question?
A very beautiful story, Jay. I have to ask, how much the short peptides do you need to see that effect, stimulating motility? That's question 1. Number 2, I know you mentioned why, but you did not explain why.
That's a great question. I guess we'll get this from the reviewer. I think it's when we use is 100 nanomolar is enough when you use synthetic peptide. However, but I don't think in real place it will be reach that high. 100 nanomolar is not that high, but when we actually find some other factor in the supernatant can reduce to 1 nanomolar. We don't know that compound yet. So we have another story. As said, you guys need to invite me back to tell you more.
Okay, but that peptide, you show that it binds to the palm A, the inner surface. Does it stay there, or does it get transported?
Right. So palm AB is it's an inner membrane protein, right? So, but because it's really short, so probably I think it would diffuse it. But we don't know how it binds. How it binds would change the conformation of the structure. We don't know yet. We have a collaboration with people who did this cryo-EM structure. We're doing that now. So we don't know.
Okay. Well, can I ask one last question? For Vibrio attachment to colonize, the field thinks it's the pali that plays the major role instead of the flagella. Is that still true? So for Vibrio colonization in animals, you need the type 4 pali, right? So then have you checked when you add this peptide, does that affect the pali expression?
Right. We did RNA-seq, we did all the measurement, and it does not affect all other virulence genes. So that does not affect the motility genes. It's post-translational kind of interaction. So.
Okay, yeah. Thank you very much.
Thank you, Jay. I have a small question. Usually when we get diarrhea, our gut should be dominated by gram-negative bacteria, right? But strep should have streptococcus. They should have a lower abundance there. What if all these peptides they were dominant, overwhelming by other stuff?
Right, but in those dysbiotic ones, actually streptococcus is pretty dominant. But those you see those patients not necessarily really dysbiotic. I think it's hard to say. We don't know, but you don't really need to have high really dominant with streptococcus because it's probably just local concentration would be high enough to induce, right? So, but that's a great question.
Can Vibrio produce their own small peptide? Can do the similar chrome sensing stuff?
You mean the actual concentration of the peptide or what?
Yeah. I mean, Vibrio produce those kind of peptides themselves.
Right. So they produce nanomolar range. It's low, but it's right in the concentration we use. And as I said, we have other stuff that can further enhance the peptide activity.
Okay, thanks, Dr. Zhu. Yeah.